and we have started as well. So Pinaki, we are.
Okay, great. So good afternoon, everybody. Welcome to today's symposium on India's vaccination policy. It's a very special day because, as you all know, the countrywide vaccination has been started today and launched by the Prime Minister today morning. So we will be discussing various aspects of the vaccination policy and also to understand what vaccines are available, to analyze the proposed plans for rollout, accessibility of various vaccines, experience of ongoing vaccination in other countries, and also other prior Indian experience with different vaccines and so on. So before we start, I'll just request Professor Ramanujam to say a few words on Indian scientists' response to COVID-19.
Thanks very much, Pinaki. Welcome, everyone. Well, the Indian Scientists' Response to COVID-19 is a voluntary group which has been working voluntarily since March, reacting to various events and providing a discussion forum for scientists, as well as preparing science communication resources and addressing a lot of misinformation that has come as well, and trying to act as an interpreter of scientific data for community. After some break, we are resuming our symposia. Several of the speakers here today have talked to us in the past. They are back to talk to us. Welcome, everyone, and thank you.
Thank you, Jam. So we have a great panel today to discuss about the vaccination process that's started in India and also the experience across the world. We have Dr. Anant Bhan, who is past president of the International Association of Bioethics and an expert on global health. Professor Shahid Jameel, whom all of us know as a famous virologist and known, is currently director of the Trivedi School of Biosciences at Ashoka University. And we are delighted to have Professor Gagandeep Kang from Christian Medical College, Vellore, who is also a well-known virologist. And incidentally, we had one discussion on vaccines in which Professor Jameel was present, and this was prior to August 15th, when the proposed Covaxin was planned to be launched and so on. So we'll have presentations of 20 minutes from each of the speakers, which will be followed by some panel discussion on different points that have been covered. And then we will open the floor for discussions. So I'll request everybody to put your questions in the chat box so that we can request the speakers to respond to that. And otherwise, I'll request everybody to please mute your mic so that there is no audio disturbance and so on. So I'll request Professor Shahid Jameel to start off proceedings and tell us about how the situation stands today regarding vaccines. Professor
Thank
Jameel.
you, Pinaki. Let me share my screen, my slides, and get started.
Sure.
Just a minute. For some reason, I don't see my slides here. Just give me a minute.
Sure, sure.
Yeah. Okay. All right. So I'll basically be talking about COVID-19 vaccines in India. And let me start by this being a very special day to really recognize not just the doctors, but also healthcare workers, all healthcare workers who have not just died in the line of duty, but also served fantastically well to alleviate a lot of pain and suffering that everyone has gone through. The defining image of today has been the director of AIMS taking the vaccine this morning, as also other healthcare workers and being vaccinated. There's obviously one image that all of us would have liked to see that's missing from here. But then, if wishes were horses. Okay, so what's a vaccine? So the word vaccine really comes from the Latin for cow, and historically it's been attributed to Edward Jenner, who took cowpox material from a milkmaid and put it into a little boy by the name of James Phipps to protect him against smallpox. But this idea of variolation had been practiced for centuries in India, China, and Ottoman Turkey before European merchants took this idea to Europe. The idea is that a vaccine is a biological preparation that provides active immunity against a disease. And it typically contains something that resembles the disease-causing agent or resembles the pathogen. It could either be a weaker pathogen like cowpox protecting against smallpox, or a killed form of the microbe. Later, killed vaccines came into being largely because of the work of Louis Pasteur and others. So the vaccine essentially stimulates our immune system to recognize the agent when we get infected at a time in future. And there is a memory for this which is retained for the future. So I just want to remind you of how the immune response works. When we get exposed to something, to a pathogen, initially there is a non-specific response, which is called the innate immune response. And many times that is able to control the pathogen that's coming in, and we're talking about virus here. But then the virus is able to replicate and also induce a more long-lasting and specific immunity, which is called the adaptive immune response. And it is this adaptive immune response that essentially leaves a memory and protects us against the specific pathogen. And that's really the basis for vaccination. So when we look at, somehow my slides are not moving. Okay. So when we look at the magnitude of the response, initially there is a period of what is known as clonal expansion, in which, and this I'm talking about adaptive immunity over here. There is clonal expansion, which means that there is an increase in the number of immune cells that get activated, that expand, and followed by that, there is an effector response, meaning that B cells that mature into plasma cells produce antibodies, and T cells produce killer T cells. But then as the infection is going down, there's also a contraction in the response. So the activated lymphocytes die, but then a memory pool survives. And it is this memory pool of cells, both B cells and T cells, that get activated very quickly when we encounter that pathogen later. And that's really the whole point of vaccination, that you create a memory and you protect somebody from infection or disease that happens later. Vaccines do work, and this is a complex picture, but very simple to explain. The size of the circle essentially shows the incidence of that disease. The yellow or orange circle shows you the point at which vaccine against that disease was introduced. And you can see that when vaccine was introduced, the incidence of disease went down very, very quickly. And this could be mumps, hepatitis A, measles, chickenpox, hepatitis B, smallpox, polio, many, many diseases this has been shown. So vaccines work, and they work very well. And they are the most economical public health tool. It has been estimated that for every dollar that is spent on vaccines, it gives a return of about $44 in terms of children who grow up to be healthy adults. So one of the most economical and one of the best return on investment of any public health measure. What are the types of vaccines in development today? So if that's a representation of the SARS coronavirus, the spike protein, which is a protein present on the surface of this virus, is the one that is targeted by most vaccines. We have whole virus vaccines, which at this point are largely inactivated vaccines. So the idea here is to take the virus, grow it up to large amounts, inactivate or kill the virus with a chemical, and then use that as a vaccine. There's only one live attenuated vaccine that I know of that's under development by a company called Codagenix, but that's early in the pipeline, so we will not discuss that. The ones that have generated the most interest are genetic vaccines, particularly the RNA vaccines. And the idea is very simple. You take a piece of RNA, mRNA, messenger RNA, that codes for the spike protein. You package it in a lipid particle to protect the RNA, because as you know, RNA is a very labile molecule. And then you inject this. The idea is that when this enters a cell, the RNA is able to directly translate into the spike protein, and that raises immune responses. So both the Moderna and the Pfizer-BioNTech vaccine is based on this principle. The Zydus Cadila vaccine in India, which has entered phase 3 trials, is a DNA vaccine. And the idea is again the same. It's just that you step back one step. So the DNA in this case makes RNA, and the RNA makes protein. The other types of vaccines that we are seeing a lot are viral vector vaccines. So here the idea is to use use another virus as a vector, essentially use it something like a Trojan horse to send the nucleic acid for the spike protein into cells. So we have seen adenoviruses, like an example of chimpanzee adenovirus is the Oxford AstraZeneca vaccine, which Serum Institute is making here as Covishield. And there are other human adenovirus-based vaccines that have been made in China, in Russia, that are also in trials. Some have had emergency approvals. And there are other viral vectors that people are trying out. And finally, there are the protein vaccines, either soluble proteins or virus-like particles. The earliest example of this is a hepatitis B vaccine. In the case of COVID, there's a vaccine from Novavax, which again, Serum Institute is a partner in India for them. That's a virus-like particle vaccine. Okay, so let's look at how the two main vaccines in India that have received approval, Covaxin and Covishield, how do they work? Here is the virus. You've grown it to large amounts. You inactivate the virus. And for inactivating it, they use a chemical called beta-propiolactone, which essentially immobilizes the proteins on the surface and also inside the virus. You inject it into people, and what that does is that the antigen presenting cells that are surveying the periphery will capture this virus, process the virus, and I'm not going into the details of the MHC process. It's just that these cells will then present fragments from the virus to helper T cells, which will help raise an immune response to both produce antibodies as well as cytotoxic T cells. So here is an example of the T helper cell essentially helping the activated helper T cell, helping the B cell produce become activated, become a plasma cell, and produce antibodies. And it is these antibodies that will then neutralize any live virus that you will encounter. So that's the broad principle on which this vaccine works. Now, if we look at Covishield, here we have the same coronavirus. We have taken the gene for the spike out, engineered into an adenovirus. In this case, it's a chimpanzee adenovirus. This adenovirus is then sent into cells. It infects cells, but this adenovirus doesn't replicate because some critical adenovirus proteins are missing, so that this virus itself does not replicate in the recipient cells. However, it releases the DNA. That DNA is converted into messenger RNA. The messenger RNA produces the protein, and it is this protein that is produced from the vaccinated cell that again raises both antibody as well as T cell responses. Now, the other question that we often face is how come it used to take 10 to 15 years to make a vaccine, and suddenly in the case of COVID, multiple vaccines are available within a year. Has safety been compromised? Has efficacy been compromised? So the point is that traditional vaccine development used to take long because everything was done sequentially. And the clinical trials were quite extended. If you see the phase 1, phase 2, and phase 3 trials were quite extended, went for about 5 to 7 years. Whereas in the time of COVID, there are many processes, especially the clinical trials, that are happening in parallel. More importantly, in the traditional vaccine development pipeline, large-scale manufacturing only happened once the product was approved, because no company was willing to spend money to create a product or to make a product that would eventually not be approved. In this case, some innovative financing mechanisms, and I think Gagandeep will possibly talk more about this, has led to de-risking companies so that they have made products even before approval. And you would have noticed that Serum Institute has already made 50 million doses of Covishield even before approval came. So that's how things have been shortened. Obviously, the post-immunization period hasn't been long enough. So we don't know of the long-term effects, but at least the adverse effects have been taken care of. It's also a very historic opportunity, and possibly for the first time, that a vaccine will be used to control a pandemic. It has never happened earlier in history. So there are over 200 vaccines in development. This is the current status. About 68 vaccines are in clinical testing. And these are the different phases the different vaccines are in. 2 have been approved for full use, 8 with limited use, and there are many that are in phase 1, 2, and 3. These are the leading vaccines globally. And you will see that the Oxford AstraZeneca vaccine, which is called Covishield in India, and the Bharat Biotech vaccine are both approved for emergency use in India. And it is that emergency usage that has started essentially today, is being given to frontline workers. The vaccines that are in trial in India, Covishield and Covaxin, I've already told you that they have not completed phase 3, but they have received emergency approval in India for target groups. The other vaccines that have gone into phase 3 are the Zydus Cadila DNA vaccine. And bridging trial for the Russian Sputnik 5 vaccine by Dr. Reddy's lab. The Novavax is under consideration. My information is that these trials have not started. Beyond that, there are two other vaccines that are in early clinical development. One is a recombinant antigen-based vaccine at BioE, and the other, a very interesting product that is being done at Genova in Pune, which is a replicating mRNA vaccine. Remember the Pfizer or the Moderna vaccine, the RNA, as it goes in, it translates and then the RNA disappears. In this case, the RNA is self-replicating, so it produces more copies of itself. The idea being that you may need very small amounts to be vaccinated, and possibly a single shot would give immunity. But there is also the concern that the RNA may hang around for much longer than in the case of the Pfizer or Moderna vaccine. So interesting platform, and we'll have to see how that turns out. And finally, Bharat Biotech is also working on a rabies vector platform, which is at this point in preclinical. You are all aware that in India, the vaccine priority is to first give it to 10 million healthcare workers and 20 million frontline workers. And this is going to take about 3 months. There are roughly 3,000 vaccination points. Each vaccination point will vaccinate 100 persons a day. So it'll roughly take 100 days to accomplish this. After that, we're probably going to go to prioritized age group of those who are above 50 years of age and those below 50 years with comorbidities. The registration for this hasn't opened yet. So that people can register for their turn. Finally, just a little bit about viral variants. The original virus that came from Wuhan has very quickly changed sometime in late January, early February. And the viruses that are now spreading are different. The D614G variant. And within this variant, we now see that 3 other clades have come up. This one in orange here are the UK variants. This one in a lighter orange shade are the South Africa variants. And here in bright red are variants that have emerged recently in Brazil. We know that these variants have spread to about 50 different countries, including India. We don't know if India has any homegrown variant simply because we haven't sequenced enough. And as we sequence more, we may find that there are also some homegrown variants here. So we've gone from this virus to a vaccine, and that brought a lot of hope. Backup really has been these mutants that have developed. And the most common mutant that has emerged is this N501Y mutant. It's a matter of concern because this mutant is at the interface of the receptor binding domain in the spike protein and the ACE2 receptor shown here in green. So people are worrying that would this mutant allow vaccines, this virus to escape vaccines, especially because neutralizing antibodies are made to the receptor binding domain. An early result on this mutation is available from the lab of Vineet Menacheri in Galveston, which suggests that viruses with this mutation and viruses without this mutation are neutralized equally well by COVID patient sera. My understanding is that National Institute of Virology is planning some studies, or maybe they're already doing some studies to directly address the UK variant and the other circulating in India using the uh, uh, phase two volunteer sera. And this is also possibly something that's going on elsewhere in the world. So at this point, we are hoping that the current vaccines will continue to protect, because vaccines also give a fairly broad response. It's not a response directed to a particular site. So I will end there and maybe let my other colleagues talk, and then we can come back later and discuss some of these issues. Thank you.
Thanks very much, Professor Jameel, for setting the stage for the discussions with the I now request Professor Kang to make her presentation. Professor Kang.
Thank you, Pinaki. I don't have slides, so I'm just going to
Sure.
talk about the various things that I was asked to cover. So to begin with, we keep talking about India has the largest immunization program in the world. We are very well set up to immunize the entire country. So let's take a look at what it really is. The bulk of immunization in rural areas and in poorer parts of urban areas that happens in India is really delivered by the government. We have 27 million children born every year. And of that, about 25 plus get immunized. And the immunizations are at birth, 6, 10, and 14 weeks, and at 9 months. And then when children are between 16 and 24 months, they get another dose of vaccines. So that is really up to 2 years of age. About 6 times these children are reached. And in addition, when you have national immunization days for polio, then you get 2 attempts to reach every child under the age of 5 in the country, twice about a month apart. And this has been building up and going on for a while. In addition to this, we have approximately 29 million pregnant women, and they have to receive tetanus toxoid vaccines, 2 in the first pregnancy and usually 1 in subsequent pregnancies. So we are now going from that to immunizing adults. And so far, in all the efforts that we've had, if you look at the results of the National Family Health Survey, looking at fully immunized children by age 5. So remember that this is 6 vaccinations by age 5. We find that only about 3 out of 5 children have been immunized. If you look at what the government calls a fully immunized child, which is a child who has received all vaccines by the age of 12 months, then the percentage goes up. It was over 80% for the last couple of years. And then in January of 2020, we reached our highest ever coverage, which was 90%. That means that actually in terms of unimmunized children, India has one of the largest numbers in the world. So we are, of course, then of course COVID came along and we had a significant drop in the immunization. It went down to under 50% coverage in March and April. So we have a strong program, we have a large program, we don't have a perfect program. We are now looking at switching this program to immunize adults, and we're going to do this in phases. We started today with healthcare workers and frontline workers. And healthcare workers and frontline workers are relatively easy to identify. So I anticipate that this will go pretty well if the uptake of the vaccines is good. After this, we will be looking at the age stratification. And again, here we are in a good place because with your immunization, with your election records, you are easily able to identify the adult population in the country that has a voter ID. Of course, if you don't have a voter ID, or if you're not in the location where your voter ID is registered to, then we might have a few problems in using that data for immunization. But in general, I think the plans that have been set up are really very, very good plans. And in terms of implementation, we will just have to wait and see how they go. But certainly we have a very strong immunization system that has been established, cold chain capacity that has been built, and a good monitoring system for seeing who has received vaccine and received which vaccine. It varies by states. If you try to get immunization records for children, which is something that I've spent the last 5 years trying to do, again, some states, good data, other states, not so great data. And of course, if you are a migrant, then no data at all. So lots of planning, lots of preparation, and we'll have to see how things go. Now, if we look at how India is going to fund its program and how the rest of the world is funding their programs for immunization, we know that the US has invested upwards of $10 billion in the development of vaccines and is being asked to invest even more in the distribution of vaccines, because while they made the vaccines, their system wasn't set up to deliver as well as it might have been. There are other countries, obviously, that are doing really well, like Israel, and the UK is doing a reasonable job, and many European countries are ramping up. Now, when you roll out vaccines quickly, you do encounter problems. You may have heard that there were concerns about deaths in the elderly in Norway, and that is something where having baseline data is absolutely critical to be able to handle communications and make sure that the immunization program retains its strength. So in thinking about how the rest of the world pays for vaccines, for the US, the government is going to pay. For the UK, the European Union, the governments have subscribed to or have made advance purchase agreements for many more times than their populations. So it looks like the rich world is in a good place. For India, we know that the PM CARES fund is where the initial payments are coming from. And India and all of the Gavi eligible countries, there were 78 Gavi eligible countries in the original dispensation, but that has been increased to 92 countries. There has been a commitment made from the global community that up to 20% of the population of these 92 countries, which did not have sufficient resources or strong enough immunization programs to purchase vaccines, 20% will be provided through something called the COVAX facility. Now, the COVAX facility is the vaccine component of the ACT accelerator, and ACT stands for Access to COVID-19 Tools. It is diagnostics, drugs, and vaccines. And the idea is that in each of these areas, the global community will take responsibility for ensuring that some resources are provided to every country in the world, irrespective of their ability to pay. Now, ACT was established by the WHO, by global partners, philanthropic organizations, and in the case of vaccines, it involved the Coalition for Epidemic Preparedness Innovations and Gavi, the Vaccine Alliance. Focusing just on vaccines, CEPI and Gavi have been working together on COVAX to come up with innovative financing mechanisms, and through a pretty incredible exercise, one of the largest multilateral agreements after climate change, they've gotten over 190 countries to sign up. I mentioned previously that 92 countries will get sufficient vaccine for 20% of their population through the COVAX facility. So why 190 plus countries? The 190 plus countries is to get the rich countries to sign up to the COVAX facility as well, with the idea that if many countries negotiate together to buy vaccines from vaccine companies, they can get much better pricing. So rich countries should come on board and make sure that they provide some money upfront, which can be provided to companies for their vaccine development. And then when vaccines become available, they will be distributed to all of these countries. So the money comes from rich countries, goes to companies, and in addition, after rich countries do their budget for the next year, rich countries will make additional donations that will allow the COVAX facility to buy vaccines for poorer countries. And these are pretty complex price negotiations, but the entire structure of Gavi itself is very experienced in complex financial negotiations. In addition, the World Bank and the Asian Development Bank have set up easy loan facilities for countries for the purpose of vaccine purchase. So in India, where will we be? We will have certainly the vaccines that the country will purchase, the government will purchase, but we will also get up to about 10% of our needs from the COVAX facility. Why less for India than for rest of the world? It's really because our population is so large, and 10% of India's population is practically the same as all of the other 92 countries, their 20% population. So this will benefit India, and then we'll have to think about where the rest of the country gets its doses of vaccines from. It's likely that some of the providers of vaccines that have been developed in countries elsewhere that are not suitable for our immunization program, those vaccines will come into the private market. So we could expect the Modernas, because Pfizer has decided that it's only going to sell to a government market. So when the Moderna vaccines come in, how will they be regulated? I think these are things that we will need to wait for, but it's likely that those vaccines will come over time. Now, if we think about what's changed and why are things so different? We've never done anything like the ACT Accelerator. We've never had a CEPI before. The ACT Accelerator really came from learnings from the last pandemic, which was influenza in 2009, where WHO tried really hard to get vaccine companies to donate doses of vaccines that could be given to poorer countries. But essentially, wound up with a few million doses of vaccine very, very late in the game and wasn't able to achieve its ambitions. So the idea of ACT being established early was to learn from the mistakes that had happened in 2009. And CEPI itself, which has kind of come front and center, full disclosure, I'm vice chair of the board of CEPI. It was established in response to Ebola. In Ebola, there were a vaccine that had been developed in the late 1990s, but that vaccine had not been taken forward by any companies because the market for an Ebola vaccine was so unpredictable. When Ebola finally hit West Africa, an urban population for the first time, and started to spread across 3 countries in 2014, everyone knew there was an Ebola vaccine. Everyone knew that there were monoclonal antibodies, but not how to use them. It took WHO, the UK, and the US, all of whom were fighting with each other, ultimately about 9 months to get the vaccine into arms of people. And by that time, the epidemic was waning because of infection control practices and management. It therefore took us until 2019 to get that Ebola vaccine licensed. And last year, we had another Ebola vaccine licensed. Shahid mentioned that no vaccine has ever been used to end a pandemic before, and that is correct. But the Ebola vaccines have been used to end outbreaks in the DRC in conflict zones, which is something that would have been impossible. The control of those outbreaks would have been impossible without the vaccines that we have today. So I'm going to stop there and take questions at the end after Anant. So back to you now, Pinaki.
For explaining the very complex structure of vaccine accessibility to the world across. Anant, Dr. Bhan, can you, maybe you can start now. And
yeah, thank you so much. And thank you for this opportunity by the ISRC community, the invite, and also a privilege to be on the same panel as Professor Jameel and Professor Kang. Just confirming that you can see my slides?
Yeah, we can see.
All right.
So if you put it in the present mode, then on the left.
Yeah, you're able to see it now in full slideshow.
Yeah.
Okay, great. So the idea was for me to focus on ethical considerations involved in the COVID-19 vaccine clinical trials, and also looking at the vaccination enterprise. Now, there's a bunch of things I plan to speak on, perhaps around the way clinical trials have been set up, mechanisms for fast tracking, what could clinical trial mode mean, which is one of the language used, and some of the current aspects of the 2 leading contenders, issues around compensation, long-term effects, etc. Now, one of the things I want to point out, and I think this has been something we have alluded to already by the 2 fellow panelists, is that part of the challenge is that India's health system is not in great shape. There is a lot of systemic issues with the health system. And what pandemics do is that it sort of reveals some of these underlying issues and probably tells you about some of the dirty underbelly, and capacity issues, resourcing issues, and the way we set up our systems. It also tells you something about the regulatory system, because the regulatory system is key in terms of governance, but also in terms of R&D as it happens. Now, clearly many people have made the argument that we should be looking at while we expedite things, we should not forget that you should not use that as an opportunity to forget the basics of science. And that is what would constitute research exceptionalism. and the importance of focusing on rigorous design, analytical integrity, and full reporting of trials is extremely important. And I think we should not forget that, because that is at the heart of doing good quality science. Now, coming specifically to clinical trials and vaccination programs, obviously there are multiple things we could talk about. That a few things just to flag are how recruitment is done, especially for clinical trials, how are risks and benefits cataloged. If informed consent is being taken, then are there concerns around therapeutic misconception, issues around conflicts of interest, the need for transparency and accountability. Now, if you look at the first element, which is informed consent, I think the most important area here, which we talk about, is how informed consent is done and how do you ensure quality and fidelity of the consent process. So there are three elements which I think are relevant. One is that you need to provide information and adequate information which is relevant to potential participants. The second thing is you have to make sure that they understand that information. And the third thing is that this is something that they should be voluntarily participating in. So for example, you can't force anyone into participating in a study. And I guess there is one additional element, which is documentation, which is also often a legal, regulatory, or an ethical requirement. The other element is once someone is in your clinical trial, then you have to ensure care for that participant, which includes medical care, which includes providing health information as relevant for that person, and facilitating care or referrals as required for them if should they have a need for that. But what we have seen happen, I think, is that at least in some of the trial sites, or at least one particular trial site, there have been issues with this, and this has been now highlighted multiple times in media. But maybe I'll just show us a small clip just to illustrate rather than speaking to it.
और क्या बोला था आपको? बीमार होंगे तो कोई फोन आया था आपके पास? नहीं। इससे आई तो ये आपने 19 तारीख को लगवाया है। हाँ। ये सारे पेज पन्ने खाली हैं, तो किसी दिन वहाँ से किसी डॉक्टर वगैरह का फोन आया था क्या आप लोगों का? अभी तक नहीं आया। बुलाया है उधर ही, मतलब उधर ही बुलाया है। और बीच में किसी ने पूछने के लिए कि तबीयत बिगड़ी या नहीं बिगड़ी, ऐसा कोई फोन नहीं आया। कोई
Okay, so for those of you who don't know Hindi, what she's saying is that she's a participant of the clinical trial. She didn't get a copy of the consent form. She was given a booklet which is supposed to catalog adverse events. This is to be done by the participant, but beyond that, there's not been any active follow-up with her from the site. She's been asked to come to the site, but beyond that, there's not been much that they've been in touch with her about. Another example, maybe again quickly, just to illustrate some of the issues as we talk about consent.
यही प्रयोग किया था कि वहां पर जाके बस चेक हो रहा है और वहां जाके 750 रुपए मिल जाएंगे, ज्यादा कुछ ही नहीं है। अब वहां जाके गए तो डॉक्टर कहते हैं कि आपके ये टीका इसलिए लगाया जा रही है कि आपको कोई खराबी हो खून में या ये वो हो तो ये सब खत्म हो जाएगा। तो सर इससे कोई वो तो नहीं होगा? तो वो कहते नहीं, आपको कुछ नहीं होगा, आप सही रहोगे। बस मगर जब से आया टीका लगा के तो तबीयत खराब है जब से ही।
Okay, so this was an example where the participant didn't really understand that they were signing up for a clinical trial. They were told you will get a vaccine, it will make you better, but they didn't understand the potential risks and benefits of participating, and again showing some lapses in terms of how information is provided. The other important component, I think, from an perspective is the issue around how do you choose the right sites to do research. So institutional arrangement and site selection. And you really need to focus a lot on this, because I think part of the problem is sometimes when you do expedited processes and you don't invest enough in choosing the right partners for your research, then you might end up in situations where their approach to this kind of science might be a bit immature, and they might not be focusing on quality and ensuring that they are having research, participant protection, as well as fidelity to procedures at heart. The role of the ethics committee obviously is extremely important here. The site ethics committee, as well as national mechanisms like the Central Ethics Committee for Health Research, to look at how recruitment is being done, how are they engaging participants, what kind of recruitment flyers are going out, and how do you ensure that research participant protection is maximized. But again, we have to historically look at how effective ethics committees have been in that role and where have been the gaps in that. One additional element I think which is also relevant here is rigorous assessment by independent regulatory bodies where there is no political interference. It has to be seen as something which is a non-negotiable component of good quality oversight structures, and this is also something which should be transparently described and talked about. So the problems with fast tracking mechanisms often are that it could impact scientific validity. You might not be able to sometimes look at long-term side effects. The social value sometimes might not be looked at in close detail. Oversight and monitoring mechanisms could be impacted. And the regulatory mechanisms, as we see right now, also in India, are not really set up for fast tracking. There are small components in the new drugs and clinical trials rules which talk about the potential for using emergency situations to fast track some things, but they don't go into a lot of detail, and hence that also becomes difficult to then benchmark them against. So as has been described already, you have two vaccines in India which have been undergoing clinical trials. For one of them, we have phase 2-3 safety immunogenicity data. That trial started in August, should be going on at least till March. So again, if anything, we only would have had early data for that. For the other one, they are doing phase 3 efficacy studies right now, which started in November, around a trial sample size of around 26,000. Again, this is data which should, even the earliest data for that should not be available before March or maybe end of February. And this is also evidence from the public information we have on the Clinical Trials Registry of India. Now, the challenge I think has been for most of us to try to figure out what is actually clinical trial mode, because that is what one of the vaccines has been allowed to be approved under. If you look at the minutes, most recent minutes of the subject expert committee, that provides some element of detail. It talks about informed consent form, a fact sheet which needs to be provided, and an adverse event form. They also talk about a protocol which apparently has been approved by the Central Ethics Committee for Health Research of ICMR and has been submitted to the subject expert committee. But beyond that, we don't really have a lot of details as to what that protocol refers to. For example, we know that this mode has never been used before. There is, what would consent really mean? Because all of the dry runs that we've seen do not really factor in time required for good quality informed consent. For example, if I were to do a study, I would tend to think that at least 30 to 45 minutes at a minimum is something you require for providing the counseling as part of the informed consent with a potential participant. Is that something which has been factored into the 100 participants vaccination site that they talked about? Is privacy something that they have thought about in terms of how would you offer that for potential trial participants? Is there a separate room? I haven't seen any description of a separate space for administering consent when this kind of a rollout is happening in clinical trial mode. Will there be an ethics committee providing oversight? Will there be one singular ethics committee all over the country? Will there be a principal investigator for this protocol? If not, what is the mechanism for accountability here, and what is the right adverse event reporting mechanism that they have looked at? There is a consent form which I think we have finally seen in the public domain. Again, hopefully this is the right one, but this has been posted, I think, online by a few journalists as well as health systems representatives. So I presume this is the right one. It has a screening component and it has a consent form part of it. Again, not going into a lot of detail, doesn't talk about components you usually want in the consent form around what exactly is the protocol, who is the principal investigator, if you have concerns about the study, who do you contact, etc. All of that seems to be missing elements in this consent form. Now, the challenge, I think, is going to be the fact that because of this lack of clarity, lack of transparency and opacity, and also because you have one vaccine where there is efficacy data available from abroad and some maybe early stage bridging data on safety and immunogenicity from India, and the second vaccine where you have safety immunogenicity data from India, but not yet efficacy data, and these are the two vaccine choices that you have, which have been both rolled out. And at the end of the day, as a potential vaccinee, I don't have a choice of which to take. Whichever one is allotted to the trials, to the site where I might be getting the vaccine administered, I will have to take it. There is obviously some degree of confusion which is being created, and people are asking questions. So This has just been posted today. I think Shankul is also in the audience, but I think many other people have also posted this letter from the resident doctors of Ram Manohar Lohia Hospital, asking their medical superintendent to not provide Covishield but to provide Covaxin, because they are, they are concerned about the fact that we don't have efficacy data as of yet for Covaxin. So will this play out much more over the next few days? We'll have to wait and see. But just on day one, that we are starting to see these patterns come up, tells us that it's not all fine, and there are valid concerns about the lack of this data being available. Now, the issue around compensation is really important, because quite honestly, we don't have a good functional compensation system for vaccine injury in this country. We've had issues with even kids getting vaccine associated paralytic polio in the past, and they've sort of been abandoned by the program. You don't have the government coming forward and taking care of them as individuals who potentially got a very rare adverse event, but then should have been taken care of as part of the public health response. So is this something similar we need to worry about? This is obviously something you would want as an ethical requirement, because it's rooted in justice, it's rooted in your reciprocity principle. It is something that in most cases, at least from an ethics perspective, should be something equivalent to a no-fault compensation modality. But we don't have that. Rather, what we have is some kind of bickering right now, which is happening. So you have the manufacturers which say that the government protect us from frivolous, in inverted commas, liability cases which come up, and no lawsuits should be entertained. Whereas the government says, well, even if we are rolling it out in restricted emergency use, the onus of responsibility, should something go wrong, lies with the industry. So, again, nowhere in this is the stakeholder who's probably the most important to this discussion, which is the potential vaccine recipient, and what this means for them, and are they really protected at the end of the day, given this sort of diversion of opinion between these two groups. And then the regulator, as has been par for the course, has unfortunately been silent on this issue. There is some reference to the compensation issue in the Covaxin consent form, but with Covishield, we don't really know. But one presumes that the government order sort of specifies that the company is liable. Specifically around long-term effects, I think that's another issue we need to worry about, because we only have short-term data, as I said earlier. And so we will have to be keeping a close watch through surveillance mechanisms. How well is that set up in this country, given what we know of pharmacovigilance? Are we specifically looking out for AEFIs, AESIs as well, which is something which has been more recently talked about in a more proactive manner, given the pandemic, etc. And if yes, what is the kind of things that you should be doing in surveillance, and do we have these mechanisms set up in terms of establishing causality, defining any linkage, developing harmonized case definitions, and having a national expert committee which looks at some of these issues. So I think these are the kind of things we should be looking out for. And if it's not there, then it shows that there are gaps in the way we are responding. The other element I think I want to highlight very quickly is that any regulatory decision, including of the kind that we have seen now, will have implications on both ongoing and future trials. For example, for those clinical trial participants in both the Covishield and Covaxin studies, which are ongoing, as I said, do you need to have an updated consent? In my opinion, yes. You should be going back and telling them that for the clinical trial, investigational products that you are in, there has now been a regulatory decision which has been made. It's a restricted emergency use, and it's only available for certain priority groups as of now, but you have a think about it. And if you want to withdraw yourself from the trial, especially because some of them might even be frontline health workers who are clinical trial participants, then they have a right to decide that. But is that happening? I don't know. Should there be talk about unblinding trial participants so that they know whether they're in the placebo arm or the intervention arm? What is the ethical obligation for placebo arm participants here? Now, some of these issues are things which have been tackled in discussion papers, including this paper which came out, a policy brief which came out last month, which seems to suggest that since we don't have an adequate evidence so far, that it might be good to let the trials continue, but ethically you need to inform the participants about the updates, and they have a right to decide if they want to leave the study or not. What would be the new standard of care? If there are vaccines under restricted emergency use mechanism, does that establish a new standard of care? Or and if that is true, then any future study, including all of the future vaccine candidates which will come to phase 3, actually, in fact, even the Zydus Cadila one, which is going to be starting its phase 3, can that ethically have a placebo arm? Because if you say that there are widespread availability, including, well, even if it is in restricted populations, then can you really say that the standard of care is no vaccine? Or does that, the availability of these vaccines, especially Covishield, which has efficacy data, if not from India, from abroad, does that establish a new standard of care? And if that becomes true, then you might be ethically obligated to provide a comparator arm and not a placebo arm. And comparator studies can be much more expensive. They have larger sample sizes. They can take longer to complete. So again, I think we need to have an open discussion on this. There are various perspectives. I was part of this policy brief where we made some arguments, but I think this is something which is also which should be playing out in public discussions, and the regulator should provide clarity on what their approach to this is, because there are obviously clear ethical requirements here. So I'll stop here. I have a small video which I wanted to play, but I wonder if there is adequate time. It's a 3-minute video which we had come up with just in reference to today's meeting. But Pinaki, maybe I'll wait to hear from you before.
I think you can go ahead. You can go ahead and play the video. We have time.
Okay, great. So before I do that, maybe I'll just share my screen and. Okay, so this talks about some of the dilemmas as I had tried to highlight, but we did do a small doodly video. Okay, all right. So that doesn't stick to all the facts. So there is no mask usage by some of the characters, and you might not really have a choice at one particular site of vaccination. But I thought it'd be useful to illustrate some of the dilemmas here. Thank you so much for the opportunity. Back to you guys.
Thanks a lot. Thanks a lot for giving us the broad ethical questions that are involved in this context. So what we will do now is maybe we can just
tomorrow
can
you go.
Sorry. Can I please request everybody to kindly mute yourself and not unmute? Thank you. So we'll just request the panelists to maybe, if they have any additional points to make, or I mean, there have been a lot of questions that have been coming in the chat box. If they want to say something in general related to the questions and so on, would any one of you would like to start?
Maybe one thing that has come through the chat box and people should be clear about is the difference between a vaccine that prevents disease versus a vaccine that prevents infection. The COVID vaccines that are under trial now, the trial endpoint is disease. It's not infection. So the efficacy is proven against disease. Efficacy is not proven against infection. But that should not really be a worry because many successful vaccines don't prevent infection, they prevent disease. I mean, those of you who take the flu vaccine annually would realize, just like me, that earlier you used to get a flu episode that lasted 3, 4, 5 days. Now, after taking the vaccine, your flu episode lasts maybe half a day. So that's the case of a vaccine that prevents disease. It doesn't prevent infection. And that's an important point to understand. Its implication is also there in your ability to transmit the virus. If you are vaccinated with a vaccine that does not prevent infection, then it means that the virus can replicate in your body to a limited extent, and you may be able to transmit that virus to somebody. So while you get the vaccine and are protected, it's also your responsibility to continue to wear a mask even after vaccination so that you don't transmit whatever little virus you make to others.
Thanks, Professor Jameel. Professor Kang, would you like to add anything more? I don't hear anything. Okay, maybe we can start taking some.
Sorry.
Sorry. Yeah.
I was.
Please carry on. Please carry.
I'm going to speak to Sandhya Kaushika's question. How can second generation vaccines be tested for safety and efficacy? Huge problem. We've put ourselves in a bind here by approving 2 vaccines under restricted use, whatever clinical trial mode is. So the question is not just for second generation vaccines, but for the next vaccines that are in development, what is going to be asked of them? Will we stop all vaccines after immunogenicity trials and start using them in the population? So you can't treat one vaccine exceptionally and then say, once you've established a precedent, then where is your justification for doing things differently? But actually, even if the vaccines had gone through efficacy trials and both vaccines, let's say, had been proven to be efficacious, we would have questions about how future vaccines would need to be tested. And future vaccines would require either a correlate of protection or a non-inferiority design. And non-inferiority designs, of course, increase the size of the trial hugely and also mean that the trial will become very delayed and very expensive. And we might not then get the second generation vaccines, which would be more fit for purpose for us. So everything that is in phase 3 trials now is probably all right. but any vaccines going to start phase 3 trials after this, or any vaccines that go into future testing, they are going to need comparators unless we can come up with a correlate of protection. If we do that, then we are on solid ground for licensure, which actually means that we need to go back to all the vaccine failures, which is everybody who got vaccinated and then got COVID-19 meeting the primary endpoint for a vaccine failure. We need samples from those people. We need to know what viruses they had. We need to know what their serum is able to neutralize or not neutralize. So those are going to be really challenging studies to do. There is a discussion, WHO has started a set of discussions on looking at what will be asked of second generation vaccines. There is going to be another discussion that follows next week, and I hope that we will have guidance then.
Thanks, Professor Kang. Professor Bhan, would you like to add anything more to what you presented?
Yeah, well, I mean, as Professor Kang was saying, that if we have vaccines like Moderna entering the mix, then we have to also recognize that their efficacy data so far is much higher than what we know for even AstraZeneca. And when you have a new vaccine like that, and if it gets licensure, even if it is for, say, the private market, then it goes back to the fundamental question of, does that then also become a standard of care? And if the standard of care is a 95% efficacy vaccine, and then any new vaccine trial which has to run against that standard of care will be a hugely expensive and a very, very complex trial to run, because the sample size, I think, will be much higher. Now, will there be an innovation there of the kind Dr. Kang is describing? We'll have to wait and see. But we need some of that thinking to come through now. The part of the problem, quite honestly, is that we don't really know how decisions are being taken, especially at the regulator level. There is a lack of transparency. We know the government is invested in both vaccines. In fact, both vaccines wouldn't even have reached where they have if the government was not a co-sponsor through ICMR. So it's not just that the manufacturer is in isolation and the government is in isolation, two separate stakeholders. They are in fact working together for these vaccines. So even on the pricing side, when you see these discussions, one wonders that why does the government not leverage the fact that they were co-sponsors in the R&D part and use that to get better prices out. So for the companies to then arm twist their way and talk about, we will only give you a X price point for so many doses, but not anything beyond that, is a bit strange, because if you are a co-sponsor, I would have thought that you would get some kind of a preferential rate almost at no profit. But again, as I said, part of the challenge is you hardly get to know who's involved in decisions, how are decisions taken, and why do we have language which all of us have to break our head to try to figure out when we have that being the only thing which is conveyed to the public.
Okay, thanks very much. So there's one question for you, Professor Bhan. So what are the protocols in place for any long-term adverse effects, if there are any? So what has been discussed in the Indian context?
Yeah, so I haven't seen a lot of detail. There are protocols with adverse events for adverse events following immunization, which are well written, but those are not specific, I think, to COVID-19. So I presume those are the ones which lay the basis of what they're going to do here as well. Except as we discussed now, here we have this additional layer of clinical trial mode, which potentially means that maybe they are, or at least those individuals who are getting the vaccine in that mode more intensely. But if the follow-up in the clinical trials which were happening by itself had a lot of gaps, and I hope that was clear from the couple of clips that we saw, then what does that mean if you are taking that same concept to scale? Are you really doing a good job in much more smaller controlled clinical trials that you're going to attempt at in much larger populations? The pharmacovigilance system in India, I think, is good in parts, but also has a lot of lapses, has a lot of gaps. So unless you have good quality pharmacovigilance and a reporting mechanism which is actually transparently identifying all of those events, you will not be able to do long-term good quality safety checkups, and I think that is what I worry about. So hopefully they'll do a better job. We'll have to wait and see. But again, the present experience is not very encouraging on that.
Okay, thank you. So one question that has come, and maybe this is something that people have been discussing also, is about the issue about dosage and the time period between dosages. So this is a lot of confusion that the government says something, the company says something else, and then in the UK there is one protocol in place. So there's lot of confusion in public domain regarding the dosages and also the time period between dosages. So it would be nice if Professor Jameel or Professor Kang can clarify on this.
And can I also maybe add on an additional component for both of my fellow panelists, that this whole confusion around the efficacy for Covishield, because I think this is an ongoing debate around, can you average out results from two diverse clinical trials and use that for regulatory submissions? Maybe that's good to open up now as well.
So, okay. When I think about clinical trials, you have to analyze per protocol. So whatever you had written, you would do. You should be doing that. And if we go by the per protocol analysis for Covishield, what seems to be showing up in the information sheets that we've seen in the paper that was published in the Lancet is just above 50% at approximately 4-week interval, we presume, with 2 standard doses. And then as you change the dose, as you change the duration between doses, it keeps going up. So we know that the minimum efficacy is going to be 50% plus, which meets the bar that was set by regulatory agencies. What we really should be doing, and this is where I think scientific expertise is critically important, is planning for different dosing schedules. And the way I think about it, this could potentially even be done in the course of the rollout. You could design studies that look at impact assessment, and you could vary the dose among people that are getting the vaccines and try and see how much of a difference there is. It will be an effectiveness study. It will not be an efficacy study, but it's still useful to think about, because then later you can modify the immunization program for subsequent groups based on the data that you gather early on.
Yeah, I agree with you, Gagandeep. That's a sensible thing to do, but I don't think anything like that is being looked at. We've made it very clear that it will be done as it was done in the trial. But also remember that if you vary the duration, people have to understand that protection has been seen after 2 doses. There is no information on how much protection one will get after 1 dose. So this is also something to consider, that if you increase the dose from, let's say, 4 weeks to 12 weeks, people have to understand that you're not protected after the first dose. So there are those challenges.
Thanks, Professor Jameel. So, Professor Bhan, one question regarding the Bhopal incident. So, are there follow-ups regarding compensation? Have people started discussing about this? And also, I mean, if similar incidents happen, what protocols, what actions are in place possible?
Yeah, so one of the issues is, we could do much better on our SAE response. I think, one of the emblematic issues from my perspective is, it's in many ways about the participant, yet the participant is excluded from the whole process. So you collect data about the participant, you collect their medical forms, you collect the case report forms, you collect probably all details with regards to their health. But then every decision which happens does not necessarily involve them. They're not consulted, they're not provided the opportunity for any kind of appeal. And we've seen that happen with the Chennai incident, which was linked to the Serum Institute vaccine Covishield. And we're still waiting for a final word from the regulator as to what was the causality assessment, and we haven't heard on that. With Bhopal, you have a series of clinical trial participants, many of them who are survivors of the gas tragedy, who talked about the fact that recruitment was done in a way that they did not understand that they were participating in a trial. They were not sometimes given copies of their consent form. For many of them, they would qualify under what is known as vulnerable populations under our regulations, which require audiovisual consenting, but audiovisual consent has not been taken. Medical care was not necessarily provided. And when they did report, they were asked to go and access care from private sources. So if that is how things are happening, and then the response is that the trial site and the investigator basically washes off their hands and says, we did everything as per protocol, and there is no fault, and there has been one death, but it's not linked to the vaccine. Yet none of all of these concerns are being really responded to. I would have thought this would have been a clear case for a regulatory audit by an independent committee coming in and talking to these individuals, trying to find out what happened and trying to respond and fix things. That is how you would have built confidence, because then there would have been an understanding that this is being taken seriously. But by actually not even caring about it, not acknowledging it, we are opening up concerns about how are we treating our clinical trial participants. And if clinical trial participants in a controlled setting are being treated this way, what does that mean when you go to scale in clinical trial mode? What if there are adverse events which happen? Will those be really responded to? Are we going to be serious about collecting that information and providing either medical care or compensation if there is causality established? I think that is what we should all worry about, because if we cannot protect our clinical trial participants, what hope do we have of conducting good quality studies in the future? Why should anyone sign up to be a part of clinical trial?
Anant, I had a question for you along that line. People who volunteered for a trial and those who were in the placebo group, do you think that they should be on priority to get a vaccine?
Yes, so they should certainly be, because they have by virtue of volunteering and contributing to the science of development. Ethically, yes, that would be a requirement. When is the right time to do it? I think there will be some differing opinions. For example, there is a clear ethical argument to be made that you want these trials to go to completion, because you want that kind of good quality data to be available for you to make a final decision around licensure. Because right now what we have is sort of a restricted emergency use decision, but we cannot only have a fair idea about the vaccines once they go to completion. But then in the interim, are they being provided good quality medical care? Do they have rescue mechanisms in place? But yes, obviously, I would tend to think that they should certainly be among the priority groups, because they have contributed to the science. And it would be unethical, for example, by that time, if we run out of government supply, then to expect poor participants to have to go and buy the vaccine, even though they were clinical trial participants, would be a travesty.
Thank you. So next question, it's sort of a layman question, but maybe it's useful to discuss this. So can somebody explain how immunogenicity, efficacy, and effectiveness are determined? Professor Kang, would you like to take that?
Sure. So immunogenicity is when you give the vaccine to someone and you measure an immune response. So far, practically all of the vaccines that we have are based on antibody-mediated protection. That's why measuring antibodies is what is done when you want to say that a vaccine is immunogenic in a phase 2 study. These are usually done in fairly small numbers of people, a few hundred people, so that you can assess the variability in the kinds of responses that people have. What you would expect to see is that a vaccine should induce the same kind of antibody response as is seen in a natural infection. Now, once you know that a vaccine is safe and immunogenic, you want to know, does this vaccine protect against disease or not? In order to do that, you do phase 3 trials or efficacy trials. And the idea here is to give the vaccine to some people and not give the vaccine that you're testing to other people and wait to see who develops disease. Your assumption is that if your vaccine is working, very few people or no people who were vaccinated will develop disease. And among those who were not vaccinated or given some other vaccine, they will develop disease like the general population. It's by looking at the difference between these two that you say that the vaccine really is working. And these are called phase 3 trials or efficacy trials because they are measuring the efficacy of the vaccine against clinical disease. But it's very important to understand that all trials are artificial settings. In phase 1, you're choosing people that are healthy, that usually don't have too many comorbidities, don't have lots of immune compromised people. So you'll be eliminating people for whom you think there might be a safety risk or in whom you might not get a real test of whether the vaccine works or not. Once you've proven efficacy in a phase 3 trial, that vaccine will be licensed, and then you go on to see, does this vaccine work in the real world? If I give the vaccine to the program and the program starts to vaccinate people, what will protection look like? Efficacy trials are set up to make sure that you can really measure protection against disease. But does that result in a reduction of disease in the population as a whole once the vaccine is given to everyone? Those real world studies looking at whether the vaccine is preventing disease in the real world are called effectiveness studies.
Thank you. Are there more questions? So while we wait for questions, so I just also wanted to ask, in the context of choices for other vaccines, are there? I mean, so here we have a situation where in this phase, at least in this phase, the person who is going for vaccination is not getting a choice of which vaccine to take, but at least the person can opt out if needed. So in other vaccines, in the case of other vaccines, are this question of choice there, and how does it work in that context?
So I think Anant should take that. But before Anant takes that question, I will answer Sandhya Srinivasan's question, which is to comment on whether those who have already had the disease, whether they should be vaccinated and why or why not. We have evidence now to show that there is reasonable protection against disease in people who have been previously infected. But the current advice is that whether you have been infected or not, you should be thinking about taking the vaccine. And the reason for that is that we don't know yet how long protection lasts. So if we knew for sure that if you've been previously infected, that you will be protected from disease for X period of time, it would make it easier to make a recommendation that if you've been infected, you do not need to take the vaccine. There are discussions that are going on, at least internationally, to say that at a time when vaccine supply is limited, people who have been infected before could defer their vaccination for a few months. The period is a maximum of 3 months at the moment. But I think this is something that we should be thinking about and keep tracking the data on vaccines and on infections. Over to you, Anant.
Sure. So, I mean, as of now, I don't think you have a choice anywhere in the world. You there are only limited number of doses available. So it's either whichever vaccine is made available, because most of the rollout is happening through usually government sources. In India, you don't have a choice between the vaccines. You have a choice of opting to get the vaccine or opting out of the vaccination. And even as a corollary of that, or as a sub part of that, there is no guarantee as of now that should you opt out right now, that you will have a future chance to get the vaccine at a later date. The other complicating part here is going to be if there is a private market which opens up. And as and when that happens, because there are a lot of manufacturers, including Serum, which has been very openly advocating for their eagerness to enter the private market. They've already talked about a price point for the Indian market. Then, it will throw in another ethical issue, which is to say that if I am a poor person, I will have to wait for my turn in the queue as per the government prioritization list. But if I am middle class or upper class and I don't mind paying 1,000 rupees per dose, I could jump the queue easily. And is that something which is societally acceptable? That by virtue of having an ability to pay, you can jump the queue, while others have to wait their turn. And I think that will be certainly something which should be worrisome. So yeah, so no choice as of now, certainly.
Thanks, Anand. One question from Facebook. So, one second. What is the current status of FDA-approved mRNA nanovaccines like Pfizer and Moderna? Do data suggest that nanovaccines will lead to long-term immunological memory relative to other vaccines developed using classical platforms? Professor Jameel, would you like to take that?
Well, I'm not aware of any long-term data. Well, there is no long-term data. I mean, the whole disease has been a year old, and vaccines much less so. But I can say that RNA vaccines are an interesting platform because you can make RNA quite easily compared to making recombinant viruses or making proteins. So tomorrow, if let's say a variant that emerges that cannot be dealt with the current vaccines, it should be fairly easy to make RNA-based vaccine against the new virus. But as far as long-term data is concerned, I don't think there's any available.
Thank you. Okay, one more question. Oops, there are too much traffic on the question, so it's just one second. So again, from Sandhya, let me take this question. What are the challenges in developing universal corona and flu vaccines?
Well, if you look at all the coronaviruses that infect humans, the sequence similarity is only about 50%. So whether a vaccine made against the, let's say, the spike protein of one virus cross-neutralize the other virus is, and whether it does it efficiently is unlikely. It would be possible if we had live attenuated viruses, because these viruses would essentially replicate and give rise to better cytotoxic immunity. And cytotoxic immunity essentially kills virally infected cells. And there may be better conservation in the cytotoxic epitopes across coronaviruses instead of the neutralizing, the antibody neutralizing epitopes. As far as flu is concerned, any flu vaccines that are based on the H and N proteins, again, because of the variability of these proteins among different types of flu viruses, they will not cross-neutralize efficiently. So those people who are working on universal flu vaccines are using other proteins from the flu virus. to generate universal vaccines. So those are my thoughts on it.
Okay. So one question from Shankar: what is the rationale for 50% being the minimum required efficacy?
Okay, maybe I can take that. So the 50% was actually decided at WHO initially, and this was part of the target product profile for vaccines. What was a vaccine that made public health sense? And we thought about it, and a 50% vaccine would be sufficient to reduce disease in populations significantly. So the minimum was set at 50%. We were actually, so if you look at how WHO sets a framework for deciding vaccines, and companies, I think, do this as well. Lots of agencies do this. You have a minimum profile, which is your target product profile. This is what we are willing to accept as a product. And then you have something called preferred product characteristics. This is what my ideal looks like. And I think we were not very ambitious with the ideal, because we said our ideal was a vaccine that would have the efficacy of over 70%. And now 70% looks pretty low to us. So I think, but there are other parts of the preferred product characteristics that have not yet been addressed, like single dose, out of cold chain, able to give it to pregnant women, all ages, long duration protection. So lots for us still to do.
Well, yeah, I mean, hindsight is always 2020, isn't it? I mean, if we were going to decide that today, we would possibly decide differently. But at that time, it was perfectly fine. Remember that before COVID came, we were taking 10 years to make a vaccine. So I think 50% is just good enough because it'll reduce disease in the population. You're essentially trying to kill a pandemic with a vaccine.
Okay, I think we'll take a couple of more questions before we wind up. So one question, again, general question: is the second infection of COVID-19 showing more severe symptoms?
Actually, no. So there is a tracking system for reinfections that have been identified. And most reinfections are asymptomatic or milder. There have been occasional reinfections where the second infection has been more symptomatic than the first, but then we don't know enough about the first infection to be able to characterize that well. So it may happen, but I see that as being really quite rare events.
So maybe this is the last thing that we take up. So given that we have so much of confusing messages that are coming out, it may lead to vaccine hesitancy. So in that context, how shall we address this through proper communication, especially for science communication groups? How can they help in this approach, and also intervene in terms of governmental, how to talk to government regarding this? Maybe all of you can share your thoughts on this and we conclude on that.
Okay, for me, I think the government made the job hard.
Okay.
So, you know, we were doing really well. We were doing well with communications. We were doing well with pathways. We had what was a very reasonable, globally lauded strategy for vaccine development. Our companies were doing a great job of developing vaccines, and they could have done more with transparency. They could have done better with their management of clinical trials, all things that Anant has highlighted. But in terms of the pathways that were developed, those were reasonable pathways for vaccine licensure. Now I never know what to say. But I think as a scientific community, we have to think about what we should be doing so that we don't lose confidence in vaccines. All the products that we have the potential to be fantastic products. We don't know that yet, but that's not gainsaying the fact that they may be absolutely fabulous and may be exactly what we need for controlling the pandemic. But how do you communicate that vaccines are a good thing, right? That vaccines are likely to be one major part of the solution for the pandemic in this kind of situation. It's a very difficult balancing act to do.
Professor Jameel, would you like to add anything?
Yeah, I would simply say that vaccines are a product of science, and science is a system, science is a process. Science also depends upon data and evidence. Science is not a system of belief, that's religion. So could we deal with vaccines as a product of science and not as a product of a belief system or nationalism or whatever it is? Uh, and communicate as a community, communicate to people that science in the long term has answers, has solutions, but science is also wrong. Science is based on what we know today. Tomorrow it may turn out to be wrong. So science never has absolute answers for anything, and there is always a margin of error. So that uncertainty, that error needs to be understood. Uh, Beyond that, I think transparency is important because transparency builds trust. Uh, Imagine if the prime minister and all the chief ministers got vaccinated today, wouldn't it raise trust in the vaccines that are being put out? Certainly very nice for the director of AIMS to get vaccinated in full public view, but it's not the same thing. And thirdly, I would say, as far as transparency is concerned, things should not be a black box. Do any of you know who the members of the subject expert committee are who made these recommendations? It's not in the public domain. Don't you and I have a right to know who's approving the products that you and I are going to use? So let's become a little more transparent and not deal with everything as a black box that it's I'm saying that this is right. Our experts are saying this is right, so it must be right. I think that attitude is going to lead us into trouble.
Thanks, Professor Jameel. Professor Bhan.
Yeah, not much to add. I think both Professor Jameel and Professor Kang have sort of highlighted the key issues. Transparency, to my opinion, is key. Because that is actually at the base of what trust exists in existing vaccine programs, future vaccines, and the current vaccines. You cannot take COVID-19 vaccines in isolation and come up with structures which are in contravention to your own regulations and guidelines. Because then what kind of a precedence are you setting, and what does that mean for the larger scientific rigor of work that we do? We have excellent scientists, we have excellent innovators, we've done some great work. Why should we potentially put that at risk in terms of public confidence in that and trust in that by virtue of of putting maybe other objectives before scientific objectives? And I think we have to be all cognizant of that and worry about that. So hopefully there will be some learning from this, and that will be seriously thought about, and we'll start to see positive changes.
Thank you. Thanks, Professor Kang, Professor Jameel, and Professor Bhan for this wonderful discussions. We are glad that you accepted to speak at very short notice. and which also happened to coincide with the special day in India. So thanks a lot, and hopefully we have been able to address some of the complex questions involving vaccination program on COVID-19, and hopefully we'll have further discussions in future. Thank you. Thanks very much, everybody.
Thank you.
Thank you for having us.
Thanks for also answering all the questions patiently on chat.
Yes.